Okay, thank you, Dan. That was great. So you've probably figured out that we're going to be having questions at the end if time permits. So I'm going to move on and introduce our next speaker. So our next talk is going to be given by Marlene Behrmann. Marlene is a professor of ophthalmology at the University of Pittsburgh. Her outstanding contributions to cognitive neuroscience have been honored by numerous awards, including her election to the National Academy of Sciences in 2015. The title of Marlene's talk is At Least 100 Years of Hemispheric Lateralization Studies and Still Going Strong. Please join me in welcoming Marlene.
Thank you. Let's make sure that this works. Thanks, Patty. I'm honored to participate in the symposium alongside my august colleagues, and I'd like to thank the organizers for this invitation. I recognize that I'm standing here today on the shoulders of giants, and I also recognize that this work would not have been possible without the contribution of many graduate students in my lab, postdocs, my faculty colleagues, the funding from NIH and NSF, and I'd like to acknowledge that I'm a founder in a company called Precision Neuroscopics. The overarching question that I will address today concerns the organizational principles that govern the relationships between the brain and behavior. One theoretical perspective argues for specialization and segregation of function by specific area, whereas an alternative theoretical account is one in which behavior is mediated by more distributed and widespread cortical circuits. This debate has raged for a couple of hundred years by now and has humorously come to be called the debate between the palpators, such as Gall, and the bird brain ablators, such as Lashley, with the perspective favoring a more equipotential or mass action view. In today's talk, I want to examine these questions concerning the principles of organization in two particular ways. In the first, I'm going to be studying and telling you about the segregation of visual function between the two hemispheres, and then I will turn to examine segregation of function within a single hemisphere in the standard two visual pathways account. I will present past data, present data, and I will also make some comments about going on into the future. Let me start with hemispheric specialization. As you undoubtedly know, the long-standing view of the hemispheres, captured well in this image, is that the right hemisphere is holistic and creative, as depicted here by these colored flowers and kite flying that's going on, whereas the bleak, monochrome left hemisphere is reflected by the faceless people working analytically in their small cubicles. Here's a somewhat more realistic depiction, ascribing neural processes to the hemispheres, most notably word processing to the left hemisphere and face recognition to the right. Fortunately, we don't need to rely on images from the popular press. Hundreds of studies have now mapped the topography of hemispheric representations by having participants line the magnet and displaying stimuli from different categories, such as words, faces, objects, etc., and measuring the magnitude and the site of the selectivity of the bold responses for these different categories. My focus today will be on the lateralization of face and word recognition, as shown in some of these functional imaging studies with the binary segregation of faces to the fusiform face area and words to the visual word form area in the left hemisphere. Notice that I've labeled these regions with inverted commas, and I will really just use these as shorthand because we know that these areas respond to a wide variety of visual inputs. I also need to caution you here. Sometimes the right hemisphere is on the left of the screen and sometimes on the right of the screen. I've been unable to keep it standardized because I'm showing other people's data as well, but I've always labeled the hemispheres. Humans have expertise for both face and word recognition. 20-year-olds recognize 10,000 faces with ease. Mature readers read 260 words per minute. And so fine-tuning or optimizing independent processes for face and word recognition may indeed be a parsimonious solution to the brain behavior correspondence problem. There are also good theoretical arguments for the segregation of representation. Faces and words are so different in their image statistics that it seems unlikely that there could be common areas of the brain that underlie both of these functions. Also, from an evolutionary point of view, face recognition has been around for thousands of years, whereas word recognition in society is much more recent and has only been popular in culture for the last 150 years. And in terms of acquisition, whereas face recognition seems to be acquired incidentally by children from very early in life over the course of development, word recognition is typically acquired in pre- or middle school with explicit instruction and drill. And the empirical evidence for segregation is obviously strong. Focal lesions to the right and the left hemisphere give rise to neuropsychological deficits such as prosopagnosia and pure alexia. There is a tremendous amount of electrophysiological data that support the claim that there is greater, let's see if I can get this. Wow, this is so far, I can hardly even, that there is greater activation or electrophysiological signal over the right hemisphere for faces and over the left hemisphere for words. And finally, from the advent of functional neuroimaging studies, we see the selective activation for faces over the right hemisphere and for words over the left hemisphere. But if you step back slightly and scrutinize the data very carefully, you can see that in almost all of these studies, there is bilateral activation. So yes, it is true that there is greater activation in the right hemisphere for faces, and there is also greater activation over the left hemisphere for words. But notice that there really is bilateral activation across many, many studies. Now, these studies typically either show faces or they show words. But in a study a number of years ago with Uri Hasson, Rafi Malach, we showed participants in the magnet both faces and letter strings. Depicted here is the inferior inflated surface of the brain from the ventral view, and on the sides are the flat maps. Letter strings are in purple, faces are in orange, and I'd like to draw your attention to the fact that both hemispheres have both orange and purple. But the right hemisphere has actually more orange than purple, and the left hemisphere has more purple than orange, suggesting that there is bilateral activation with weighted asymmetry to the left and the right hemisphere. And interestingly, if you take the peak coordinate of the activation for faces and words in the right and the left hemisphere, the coordinates are in fact very similar to each other. So where does this bilateral weighted asymmetry come from? We have articulated an account where this emerges over the course of development. So in very young children, before the acquisition of literacy, both hemispheres are becoming increasingly tuned for face recognition. However, once children start going to school and start learning to read, the acquisition of orthography gives rise to greater optimization for words in the left hemisphere, and by virtue of this, face recognition gets shunted out primarily, although not entirely, to the right hemisphere. So there's bilateral activation, and the word recognition comes to be optimized in the left hemisphere because it needs to cooperate with language areas which for the majority of the population are left hemisphere dominant. Last, in adult, we have the full-blown weighted asymmetry. So let me show you some evidence that supports some of these ideas for coordination and competition in cortex. We've recently finished scanning 55 individuals, and this is the group average map of those individuals on flattened surfaces. Words are in blue, faces are in red. Both hemispheres have got blue and red. And notice that there is this weighted asymmetry with more blue for words in the left hemisphere, primarily co-localized with Broca's area in the left hemisphere, and more red than blue in the right hemisphere. And this has suggested to us that there is both cooperation, as shown by this green bracket over here, as well as competition between the hemispheres, with faces and words being pushed out, albeit not exclusively. We can also look at the correlation between words and faces in these 55 individuals, and you can see that the greater the sum selectivity for faces, shown here on the y-axis, so the greater the sum selectivity for text on the x-axis. And this very nice correlation holds. For those who are interested, I would draw your attention to a paper that we published in PNAS last year, in which we instantiated these ideas about competition and cooperation in a convolutional neural network with topographic organization. But you may be wondering, if it is the case that both hemispheres have both face and word capabilities, why would we ever see neuropsychological deficits? In fact, if focal lesions like this should just lead to the recruitment of the preserved hemisphere stepping in and compensating. We looked at the individual profiles of these 55 individuals, and indeed the majority of them adhered to the pattern of bilateral activation. But there is a really small minority who have unilateral activation. So faces entirely in the left hemisphere, words entirely in the right hemisphere. I would conjecture that these individuals are going to be our pure lexics and prosopagnosics of the future, should they sustain brain damage. And indeed, pure alexia and prosopagnosia are really rare as well. How fixed or malleable might these kinds of hemispheric organizations be? Well, I've already suggested to you that in focal lesions in adulthood, these kinds of neuropsychological deficits emerge, and in fact, they are really difficult to rehabilitate. But looking at these questions in adults is probably not the right model to be exploring. What we really want to do is we want to look at the model where maximal plasticity is available, and that is in children. So I'm going to show you some data from 39 children who've had extensive resections in childhood because of drug-resistant epilepsy, either 24 left hemispherectomy and 15 right hemispherectomy, and we're going to look at whether or not there is recruitment or malleability in the single preserved hemisphere. The experiments are very simple. There's a fixation point, and then a stimulus comes up, and another fixation point, a second stimulus, and then they have to respond whether the two stimuli are the same or different, and they complete one set of blocks for words and for faces. And here are the data. Accuracy is on the y-axis. The blue blob is the control data, with each of the light blue dots being the younger controls and the darker dots are the older controls, because we sex and age match to the hemispherectomy cases. And notice here that, and sorry, I should tell you, these are patients. Oh, good, you can see this. These are individuals with a preserved left hemisphere. These are the individuals with a preserved right hemisphere. On average, and independent of which hemisphere is preserved, these patients are scoring about 85% accuracy, statistically inferior to the 95% in the controls, but perhaps dramatic, considering that 50% of their brain has been removed. And if we look at the data from the word condition, we essentially see the same, something like 85% accuracy, statistically poorer than the controls. And so these data lead us to conclude that these patients who perform at about 85% can do so irrespective of which hemisphere is removed. Moreover, their performance for faces and words within a single developing hemisphere is correlated, and so faces and words can emerge equally in a single preserved hemisphere, attesting to this plasticity. Okay, so this brings us up to the present. Let me tell you a little bit about the future. And I'm not going to show you any data, because if I do, that would mean it would be the present. So I'm only going to show you glimmerings of, of the findings. Okay, there are many ongoing questions that we have about the brain-behavior relationships. Questions about neural representations, the underlying neural code, temporal dynamics, computations. And we need much more fine-grained spatial and temporal resolution to begin to look under the hood and address these kinds of questions. So we have just, in the last couple of years, been collecting electrophysiological data from children who have intracranial depth electrodes implanted as part of their pre-surgical evaluation for mapping prior to the surgeons going and resecting whatever area is associated with the epileptogenic focus. Shown here are 4 different slices. Each of these lines is a single electrode. There are about 14 to 18 electrodes that are typically implanted, and on each of these electrodes there are something like 14 to 16 individual contacts. I'm going to show you just a little data from this particular electrode, which traverses the inferior temporal lobe, in fact, the sort of very regions that we're interested in for face and word recognition. And for illustration and sort of tantalizing clues, the data that I'm going to show you are changes in broadband versus the baseline. So on the x-axis here is time. This is the pre-stimulus baseline, and this is the activation. And on the y-axis are each of these individual contacts along the shaft of that electrode. Participants view blocks of faces, places, words, objects, and scrambled images. And I'd like to draw your attention to the fact that perhaps most obviously depicted here is this nice rhythmic response which basically follows the presentation of the stimulus. So faces are on for 800 milliseconds, off for 200, on for 800. So we see the frequency recapitulation in broadband of the experimental design. We can, of course, calculate things like the mean percent change in power. These are just for the contacts along this particular electrode, and you can see that there is a significant change in power, particularly in this electrode for objects, and you can see it most obviously depicted in this very clear example here. Okay, so I think we can now begin to pull together some ideas about brain behavior relationships with respect to hemispheric organization. It seems to not be binary, but rather more graded and weighted. There are individual differences. The hemispheres appear to be really highly plastic in their ability to evince function. And we are just beginning to get a look into more complex and highly resolved issues concerning neural code and the temporal dynamics, and this is very much the trend for the future. Okay, I'm going to switch over now and address a conceptualization of within hemisphere organization rather than between. So historically, as I'm sure you all know, one of the seminal studies in the whole of visual neuroscience done by Mishkin and Angelide and colleagues had monkeys learn 2 different tasks. The first task is a shape or object discrimination. So the monkey needs to learn that the peanut is situated in a food well under this pyramidal object. And it makes no difference what the spatial location of the object is. This is how they access the peanut. In a second task, the shape itself was no longer predictive of where the peanut would be found. The only way that the monkeys could learn to find the peanut was because one of these 2 pyramids now was in close proximity to the cylinder, and that indicated the location of the peanut. They then went on to lesion some of the monkeys' temporal cortex and other monkeys' parietal cortex, and showed a double dissociation such that after the temporal lesion, the monkeys had lost their ability to do the shape discrimination task, but had preserved spatial localization performance. And in complementary fashion, those who had a parietal lesion had preserved shape representations, but performed poorly in terms of spatial location discrimination. And this, of course, gave rise to now the very well-established ideas about division of labor in the cortical visual system, with a dorsal Ware system and a ventral Watt system. More recently, this distinction has undergone a reconsideration, most obviously by Goodell and Milner and colleagues, who've argued that dorsal cortex plays a role in vision for action and suggested that the word how might be applicable. Irrespective of how you think about dorsal cortex, what I want to do now is evaluate whether or not there is any what information in dorsal cortex. Is there any object perceptual capabilities in dorsal cortex, or is it really the case that this is under the sole purview of the temporal lobe? So in one imaging experiment, we showed the participants either full objects, this is a little clock, and we systematically then decomposed the intact display into 4 parts, 16, 64, 256, using a box scrambling method. And we then characterized every single visually active voxel in terms of its slope across these 5 parametric conditions. So if you notice here in early visual cortex, these are in beta weights. The highest beta weight is for the most scrambled condition, and it becomes incrementally decreased as the stimulus becomes more intact. And undoubtedly, in early visual cortex, the presence of these extra edges and the high spatial frequency is going to induce activation in early visual cortex. Notice here, from the ventral perspective and also from the dorsal perspective, there is much red. And red here indicates the beta weights that show a positive linear regression from the least scrambled to the most intact, indicating shape sensitivity. So the more intact the object, the greater the response, both ventrally and dorsally. And we repeated this experiment using a slightly different manipulation called diffeomorphic scrambling, in which we took the full intact object and we gradually just wrung it out and altered its shape. And the results are essentially the same, somewhat weaker in the diffeomorphic scrambling, but a very good replication. And we did multidimensional scaling across the slopes for many of these different ROIs, both dorsal, shown here in blue, and ventral in orange. And you can see that in some locations in this multidimensional space, dorsal and ventral locations are extremely similar to each other. But you could be asking, well, what exactly is dorsal cortex doing? What does it bring to the party? In recent imaging studies, we've been examining this question, and we started off by identifying regions in dorsal cortex that showed significantly greater activation in determining object-centered parts. So, for example, these stimuli have all got 2 parts on a vertical stick. The 2 parts are always the same, but they have different spatial relations, and contrasted that with a feature design in which the features all differed across the display, but they all shared the same spatial positions. So we identified 4 dorsal regions in posterior and anterior intraparietal sulcus, as you can see over here. And then, using independent data, we pulled out the voxels that show the greater sensitivity to part relations, but we also looked at sensitivity to other functions that we know are subserved by dorsal cortex, including distance in this allocentric spatial localizer. We have depth localizers, we have tool localizers. And what I'm going to show you here is just the selectivity for part relations in dorsal cortex, having stripped away all of these other functions. And I hope you can see here, with beta weights shown on the y-axis, that in all 4 of these regions, there is significantly greater activation in the subset of voxels for part relations than for any of these other functions. But how does this play a role in object recognition? So these same participants completed a second experiment in which we showed them 5 exemplars from 5 different categories controlled for lighting, color, or any other very obvious sort of low-level statistics. This is an event-related design. And we went on to show decoding using SVM decoding with tenfold cross-validation. What I'm showing you here on the y-axis is the decoding accuracy. And we see significantly enhanced decoding activation in some of these dorsal regions, particularly in the right posterior intraparietal sulcus. But I know that you're probably thinking, well, that's not surprising because dorsal and ventral cortex are really tightly coupled. There is massive interconnectivity. And so dorsal cortex is simply inheriting the propagated signal from ventral cortex. And that's what drove our next question. What are the temporal dynamics of object recognition in dorsal and ventral cortex? And we took advantage of a fantastic set of data published last year in J-Neuro by Garari, Kaplowitz, and their colleagues, in which they showed participants 4 different categories and collected very high-resolution ERP data. We used various kinds of atlases, both to identify regions of interest in dorsal and ventral cortex, and then we used sort of acrobatics in order to be able to identify exactly what the corresponding electrodes are, and then did decoding over time in the dorsal versus ventral electrodes. And I'd like to just point out to you this very specific point, and that is significantly greater earlier decoding in dorsal cortex than in ventral cortex. And we see it here both in the earlier peaks, and we also see it here in somewhat later peaks, suggesting that dorsal cortex is likely to be independently computing object properties without relying on ventral cortex. And this brings us up to the present. So let's move on to the future. So we're also conducting intracranial recording experiments in parietal cortex in children with pharmacologically intractable epilepsy. We repeated the same task of an event-related design, and the question here was whether or not from dorsal electrodes we could both decode category and then we could decode individual exemplars in each of these subsets. Here is the broadband. This is the mean percent power change. This is the baseline condition. This is the time post-stimulus onset. And you can see that in this post-stimulus onset, we can see peaks associated with each of these different categories at different points in time. And we also have now begun to do the exemplar decoding, and it turns out that we can actually do exemplar decoding in dorsal cortex as well from these intracranial electrodes. So let me wrap up here by telling you that I have addressed 2 long-standing conceptualizations. The first is that the hemispheres have segregated and independent visual functions. And the second is that within a hemisphere, there is segregation of function into dorsal versus ventral cortex. To return to the overarching question, the current and perhaps future data seem to be hinting towards an idea of brain behavior organization, which is more consistent with an account of graded, distributed function of integrated circuits, some of which can be widespread across cortex. And so it seems that the debate is being held at present by the bird-brain ablaters. And more recently, the terms that are being used for this kind of debate is between the splitters versus the lumpers, where the lumpers are favoring an idea in which neural circuits, even widespread, are well connected and can support complex visual behavior. So I will once again thank the enormous contribution of so many people, so many funding agencies, and I'm very excited to be present at this auspicious occasion. Thank you.